So good afternoon, everyone. So first, good afternoon to our participants in Asia. Good morning and good evening to old and new friends in Europe and US. I would like to first express my sincere gratitude to RUIS and the webinar organizers, particularly Karina and Anna. As you know, I'm an tumor immunologist and a beginner in the field of viral immunology. I would like to thank Professor Olivera Finn and Professor Faith Oshia, who encouraged me to share my opinion on this interesting topic. Unfortunately, Professor Ye could not connect us. I need to be my own moderator. So I start with my self-introduction. I received my PhD degree in immunology from Paris 11 University, followed by postdoc training in INSEM, Gustave Roussy Institute, and Cordelier Research Center in Paris. To extend the pursuit of research in tumor immunology, I established my own laboratory in Suzhou Institute of Systems Medicine, affiliated with Chinese Academy of Medical Science since 2015. My research focus is to decipher how multi-level stress response, particularly therapy-induced cellular stress response and environmental stressor-induced psychological stress response, modulate immune reactions. With the approaches of systems medicine, we aim to explore how various forms of stress response and cell death modalities, such as autophagy, apoptosis, necroptosis, ER stress, pyroptosis, modulate the immunogenicity of cancer cells, as well as the immune contexture within the tumor microenvironment. In addition, my group in Suzhou aims to dissect the complex circuits that link psychological distress to deleterious perturbations in the cancer immune dialogue. Right now, I took the role of Immunology Platform Director and Dean Assistant overseeing external communications and collaborations. I'm also serving as a reviewing board member of Current Research Immunology and a review editor for eLife. So, okay, without further delay, I would like to share some of my opinions on the impact of psychological stress on virus-associated immune responses. Can I have my slides, please? Thank you very much. The pandemic outbreak of coronavirus disease 2019 is caused by a highly contagious virus, officially named as SARS-CoV-2. The reproductive number R0 is an indicator of how contagious a disease is, meaning how easily it spreads from person to person in the community. According to different mathematical models, estimated R0 for COVID-19 is between 2.2 to 5.7. It is a bit higher than SARS, MERS, Ebola, and influenza. COVID-19 usually initiates from a long and asymptomatic incubation period, which is called stage 1, with or without detectable virus. It evolves to stage 2, showing mild to moderate symptoms, such as cough, fever, and fatigue, with detectable viral load. Without active interventions, it finally enters stage 3, exhibiting severe symptoms, such as pneumonia, multiple organ failure with high viral load. In contrast to other less contagious viral infection, COVID-19 brings a huge crisis to our healthcare system due to the exponential growth of confirmed cases and much delayed increase in the recovery cases. As we can see that, to better understand the challenges and find out the countermeasures of this novel infectious disease, let us take the epidemiological data from China, for example. The upper left panel shows the curves of cumulative or existing confirmed cases, recovered cases, severe cases, and cumulative deaths in China between January and March 2020. Luckily enough, as you can see that the red line crossed the green line in the halfway, thanks to the strict lockdown policies that has been launched on January the 23rd, when exponential growth of confirmed cases just started. COVID-19 brought us huge challenges due to the following facts. First, it has a long and asymptomatic incubation period. Therefore, large numbers of asymptomatic coronavirus carriers are very difficult to be identified. It causes a high rate of hospitalization, and currently there are no effective medications or vaccines. Antibiotics generally don't work. Antiviral drugs, such as remdesivir, may accelerate recovery to some extent, and the role of anti-malaria drugs, hydroxychloroquine, is still under debate. So right now, only palliative or supportive care interventions are available. And most importantly, the recovery period is usually long. It can go up to weeks and months, causing shortage of medical resources. So right now, the most effective counterparts against coronavirus 19 include cut off the chains of transmission, like wearing masks, lockdown strategies. And since there are no drugs or vaccines available, we're completely dependent on the proper functioning of our immune system to go through this disease. And the development of drugs and vaccine are urgently needed. Next slide. So of course, although we rely on the immune system to fight against COVID-19, it doesn't mean the more the better. The antiviral immunity and inflammatory response need to be balanced. SARS-CoV-2 can infect cells via the surface receptor ACE2 and TMPRSS2. And the active replication and release of the virus can cause perturbations of host cells and the release of damage-associated molecular patterns, which can be detected by the neighboring epithelial cells, endocellular cells, and macrophages, which can trigger the generation of pro-inflammatory cytokines and chemokines. And these soluble factors can attract monocytes, macrophages, and T cells to the site of infection, potentiating local inflammation via positive feedback loops. So in the context of a healthy immune response, the initial inflammation can attract NK cells and virus-specific T cells to the site of infection, where these cells can eliminate the infected cells before virus spreads. Neutralizing antibodies in these individuals can block viral infection. Macrophages can recognize and neutralize the virus and the apoptotic cells and clear them by phagocytosis. However, on the contrary, dysregulated immune responses usually display excessive infiltration of monocytes, macrophages, neutrophils, and NK cells. These will normally trigger overproduction of pro-inflammatory cytokines, which eventually damage the lung infrastructure, and ultimately the cytokine storm leads to the multi-organ failure. In addition, the non-neutralizing antibodies produced by B cells can enhance viral infection through the antibody-dependent enhancement effect. I'll go to the next slide. So very important question is why it is different individuals can show large variations in their susceptibility to and the severity of COVID-19 infection. Besides the virus-associated factors, such as the virulence, mutation rates, and viral load, the internal environment in different individuals that respond to viral infection can be the key reason. And in my mind, a careful analysis of the networks composed of different organs, tissues, the cell populations, proteins, genes, and metabolites will reveal the determinative factors which account for the individual distinctions. And importantly, we're not isolated individuals. Special attention should be paid to the external environment and the social society. Indeed, as we noted that COVID-19 pandemic can cause severe psychological stress in medical practitioners and nurses due to many reasons, such as shortage of ICUs and medical supplies, lack of effective treatments, high risk of hospital infections, the self-quarantine and social isolation, and occupational burnout. It was published in German network Open that the frontline healthcare workers directly engage in treating patients with COVID-19 are 3 times likely to suffer insomnia and more than 50% more likely to suffer depression or anxiety. And indeed, anxiety, depression, fatigue, insomnia, loneliness are frequently occurred in COVID-19 patients due to the difficulties in hospital admissions, the life-threatening disease progression showing the breathing difficulties, enduring intensive fatigue. They are worried about their family members due to the social isolation as well as the heavy medical care expenses. Still, it remains unknown whether psychological stress and their adaptation can be linked to the increased incidence, the severity, and mortality in overload hospitals. So what about healthy individuals? For healthy individuals, life is also not easy. We are confronted with supply shortages of masks, food, as well as other essentials. These can lead to the panic shopping, which further exaggerates the problem. Electronic device and social media can largely accelerate the information spreading. However, it includes both accurate information and misinformations. The spreading of COVID-19 made social distance absolutely necessary, meaning that we're not allowed to have hugs, no kisses, and no handshakes. COVID-19 also triggered financial crisis, job loss, and bankruptcy. Besides, the biased opinion and misinformation also augment the racial inequality and conflicts. So the lockdown policies are frequently applied in multiple nations, and it fundamentally changed our daily life, creating social isolation atmosphere, which is absolutely necessary to cut off the chain of transmission. So the gathering are prohibited, travels are either reduced to the minimum or completely cancelled, and essential outdoor activities are strictly controlled, and full protection is mandatory. We spend more time with our family members, creating not only joy, but it also creates family conflicts. So I've been through a work from home period for 1 month in February. I have to admit that although I love my kids very much, but my efficacy of work is heavily compromised. And the approaching of deadlines of the work is quite stressful. Due to self-quarantine, some people need medical care, and these can be largely delayed or the medical care is insufficient, which also increase our worry about the health issues. So an important question is, what are the stress mediators and how can they modulate immune responses? As you know that this stress response can create a lot of factors, which right now I give it a temporary name called stress-associated molecular patterns, meaning that it is stress mediators and it can be detected by the immune system and modulate immune responses. These stress can induce fundamental changes in the living organism, such as alternations in the neuroendocrine circuits, the metabolic patterns, and the microbial colonization programs, particularly the GI tract and lung. These systematic changes are usually accompanied with the emission of danger molecules indicative of disruption of the homeostasis. And these danger molecules, or so-called stress-associated molecular patterns, can include neuroendocrine factors, the abnormal metabolites, the metabolites mediate the epigenetic modifications, as well as the metabolite-driven protein modifications. Also, the macrobiotic changes, they can also generate abnormal metabolites, neurotransmitters, as well as other highly conserved small molecules or motifs, which we can call microbe-associated molecular patterns. So decoding the molecular mechanisms that can modulate the generation of LCMV molecules and their immune recognition will largely improve our understanding of the stress-induced immune modulation, particularly in diseases like COVID-19 infection. So we usually apply the murine models to analyze these mechanisms, as it is easier in well-controlled conditions. The frequently used model is called social defeat. By applying these models, we introduce an animal called CD1, which is in male mice, which has the breeding experiences, which are retarded breeders. And these mice are quite aggressive. When we introduce a B6 mice into the cage of these mice, it would normally launch attacks to these poor B6 mice. We let it interact for 10 minutes and then separate them with a transparent isolator. And these isolators are transparent, so they can see each other. They have holes in the isolator, so we can also smell. They can also smell each other. They can hear each other. And after 24 hours and staying in the same cages, these poor big mice were introduced to our new cages containing a new CD1 animals. And this protocol can last for 10 days. Normally, at the end of these social defeat protocols, we'll do quality control. And typically, we use a behavioral test such as the light dark box test. Without the social defeat protocols, the mice can have normal locomotion in both light box and dark box. However, in the presence of social defeat pre-treatment, we can see that the total locomotion is largely decreased, and also the movement in the light box is significantly reduced. Another test that we applied for this behavior changes is called open field, which resemble the exploratory activities of the mice. And as you can see that social defeat protocols can largely reduce the activities of the mice exploring the box, particularly in the central area. So these two behavior tests indicate that social defeat can induce anxiety. In the next slide, we also see other tests, such as splash tests. So we use 30% glucose saccharide solutions and spray them on the fur of the mice. So if the mice are not stressed, they will launch the grooming activity immediately, and the grooming time can be durable. However, if the mice have experienced social defeat protocols, they have delayed grooming launching, and the total time of grooming activity is reduced. And also by the social interaction test, we can quantify the activity and the time and total locomotions of the mice entering the interaction zone, with or without the CD1 animals in the cage. And as you can see that both splash tests and social interaction tests show that these mice show symptoms of depression. And interestingly, the downstream of stress, you have some classic stress modulators, such as the stress hormones and neurotransmitters. As you can see clearly here that the social defeat protocols can elevate the production of cortisols as measured by the levels of cortisols in the peripheral blood. And interestingly, the level of the cortisols reversely correlated with the total locomotion. We also see changes in the level of neurotransmitters, such as serotonin and norepinephrine. As you can see that the level of serotonin in the peripheral blood is largely reduced, as well as the level of norepinephrine. And the level of serotonin correlates with the total locomotion. And upstream of the cortisol production, we can see ACTH production is absolutely mandatory. And as you can see that the social defeat protocols can increase the production of ACTH, and it correlates with the emergency of the cortisol secretion. And very interestingly, we can see that the 10 days social defeat protocols can induce durable stress responses in the mice. And when we measure the total locomotions, the entry in the light box, and distance in the light box, these three parameters, before stress, immediately after stress, and 1 week after social defeat protocols, we can see that these three parameters can remain quite low at the two time point after stress. And importantly, the level of the cortisol plasma levels is increased even 1 week after social defeat protocols. We have also tested longer time point as long as 1 month, and both time point it works. So we also ask ourselves, what are the possible explanations how could stress modulate the virus-associated immune responses? Of course, we go to the literature and find some indications. Accumulating evidence suggests that sustained psychological stress can pronouncedly influence the innate and adaptive immune responses against pathological invasion. And clinical observations uncovered exposure to persistent life pressure leads to increased susceptibility to infections, which can be linked to sustained reduction in NK cytotoxic function, the virus-specific T-cell response, and antibody production. And it is known that the negative emotion and elevation in circulating cortisol levels can be associated with rapid reduction of CD4 T cells in HIV patients. And stress can be negatively shape the long-term antibody response to an influenza vaccine, rendering their increased vulnerability to viral infections. Also, literature suggests that the glucocorticoid secreted after stress is associated with immune suppression, since stress-induced surge in glucocorticoid can switch the balance of T-cell responses by impairing Th1 responses and favoring the Treg and Th2 cell responses. And importantly, glucocorticoid can disrupt the viral antigen processing and presentation by dendritic cells, leading to reduced cellular and humoral immunity against the virus. In addition, glucocorticoid biosynthesis during virus infection leads to the selective tissue-specific PD-1 expression on NK cells, which both prevent immunopathy without compromising the viral clearance activities. Also, other stress mediators such as catecholamine, particularly norepinephrine and serotonin, associated with activities of immune regulation. Catecholamine can negatively regulate antiviral immune responses, and blockade of catecholamine with beta antagonists during chronic stress can restore CD8 T cell activation against the virus challenge. Catecholamine also can disrupt the intestinal barrier, thus perturb the gastrointestinal microbiota and promoting the colonization of the pathogenic bacteria. Besides, norepinephrine can liberate ferric iron from the innate immune defense proteins such as transferrin and lactoferrin. And released ions can be used as nutrient element to support the growth of harmful bacteria. Importantly, in the RNA virus LCMV infected mice, platelet-derived serotonin can delay the virus elimination and increase the immunopathological liver cell damage by enhancing the virus-induced CD8 T cell responses. And indeed, in our previous study showing that the stress can compromise anti-tumor immunity by downregulate the antigen expression associated with the dendritic cell antigen-presenting pathway MH class 1 and MH class 2. And also, with the tumor microenvironment, the interferon gamma production by tumor-infiltrating T cells can be largely decreased, which can be found both in transplant tumors and the urine-induced the primary lung cancer models. So what about the virus infection models? So we also launched experiments in this way. We first led them to analyze the virus-specific T cell responses. We took advantage of the transgenic P14 mice, which express specific TCR for an LCMV epitope GP33. So after 10-day social defeat protocol or left untreated, and we performed the quality control of the blood test of cortisol levels and behavior tests showing the symptoms of anxiety and depression. And then at the time that P14 cells were labeled and adoptively transferred into this recipient mice, at the same time when the LCMV were injected through the intraperitoneal injection. 3 days later, spleen were harvested, and then we analyzed the antigen-specific T cell proliferation. And strikingly, what we observed is that the proliferation of P14 cells in stressed mice is significantly decreased. This is about T cells response, What about B cell-related response? And we use similar models, and this time we first perform the social defeat protocols or left the mice untreated, and then injected the LCMV virus through the intraperitoneal injections. And then we harvest different batches of mice, the spleen, and isolate the splenocytes and analyze by flow cytometry the percentage of plasma cells. And indeed, we found that after infection, the percentage of plasma cell elevated. And interestingly, at day 15, we found a large decrease in the percentage of plasma cells in the stressed group, indicating that stress can compromise antiviral plasma cell responses in the murine LCMV models. We further analyzed other parameters, such as the gastrointestinal bacteriome, and we use the sequencing strategies to analyze the differences between the stressed group and unstressed group. And as you can see that by the POC analysis, these two groups show distinct composition of the bacteriomes. And we can see that the Lactobacillus is largely decreased, while the H. pylori is largely increased. And among the top 50 abundant OTUs has been observed, we found indeed a large amount of these OTUs are significantly upregulated or downregulated, showing that a significant change of the total gastrointestinal bacterium alternations. And interestingly, we also observed some changes in the GI tract virome by first removing the virus, the free RNA and DNA from the host cells, as well as the bacterials. We focused on the DNA sequences isolated from the viruses in the GI tract. And then we indeed found significant changes between these two groups. And then we performed the association. We have found that the altered bacteriomes and viromes upon stress response. Also, we found systematic perturbations of the cytokine profiles, as well as the metabolites in the peripheral blood upon stress, with the strategy of targeted metabolic analysis. And also, we use the we use the antibody array to analyze the cytokine profiles. We found significant changes. So later on, this study has been finished. We will continue these systematic approaches to decipher these parameters, such as the stress hormones, the neurotransmitters, as well as their sensors on different immune cell populations. We'll analyze the immunome, such as those in the lung, in the GI tract, as well as in the blood, by using several new technologies, such as single-cell sequencing, the multiplex staining of the tissue, as well as the antibody array. We also use the targeted and untargeted profiling analysis of the metabolites, and also with the strategies to analyze the gastrointestinal and pulmonary microbiome, we find some huge differences. With systematic approaches and data integrating, we have hoped to find some new mechanisms explaining how this psychological stress may affect antiviral immune responses. And lastly, and most importantly, I would like to acknowledge a lot of colleagues who support this study, particularly my colleagues, Professor Aiping Wu and Professor Heng Yang, as well as the team members, Dr. Han Na, Wang Xianyue, Liu Ling, as well as Dr. Ling Xia, Li Peipei, Jin Ziqi in our laboratory. And also the metabolic analysis, we have strong supports from Professor Zhang Lihua and Zhao Qun from the Institute of Chemical Physics, Dalian in China. And also we have support from the long-term collaborations and my mentors, Professor Peter Kramer and Professor Laurent Zebogel from France. And lastly, I'd like to wish everyone stay safe, healthy, and a joyful life. Thank you very much for your attention. Now we can switch to the Q&A sessions. And let's start with some of the questions. Do you think is Dr. Sultan Akitaru Do you think physical activity or regular exercise can have effect to remove stress and immune against coronavirus 19? I think that it is really good idea to keep fit and keep joyful, to communicate more with the people around us, to share joy and sadness, and also keep fit with physical activities. I think it's really important. It can boost immune responses. And we also have questions from Nadim A. Ramdan. Does a previous infection with a member of the corona family give partial protection against infection with COVID-19? It is really an interesting question. And of course, as we have no drugs and vaccines, we completely rely on the immune system to fight against the virus. And indeed, some people do show severe symptoms, but other people can be only asymptomatic carriers. And I think it may be due to previous encountering of the virus of the same family or some cross-reactivities of the existing antibodies or T-cell responses. I think that's a question remains to be fully explored. Also, there's one question about the mode of transmission of COVID-19. Okay, I think it's, of course, a question for epidemiologistic, not for immunologists, but perhaps that you can have the transmission in multiple ways. The most classic one is through the respiratory droplets when possible. But also there are other reports say that fecal-oral transmission route is quite possible. So we don't have definite answer yet, but of course, to avoid contact or indirect contact is absolutely necessary to block the chain of transmission of the COVID-19. PNEI is my biggest love, is Maria Ivanovska. Sorry if I didn't pronounce your name correctly, forgive me. PNEI is my biggest love in immunology. What do you think? What do you think which LCMV that you mentioned can be used to measure the effect of stress on immune reactivity? Something like prognostic marker? I think it is also a very interesting question and is undergoing because we really need to monitor different changes in these so-called SAMP molecules, such as the stress hormones and neurotransmitters. The level can be quite dynamic, and the route or the place that you harvest samples to test, such as those in the blood or the urine or other tissues, you may have different results. So it can be quite interesting question that needs further investigation. And also for the metabolites that I previously mentioned is quite dynamic. It can change frequently as time goes by. So perhaps we have to define the time and also the level of it can be defined as positive or negative as prognostic markers. We also have a question from Boris Takarkovsky. Do anti-stress drugs or medications reverse immune parameters? It is absolutely a very interesting question. In our previous experiments with the murine tumor models, we do use some antidepressant, such as the serotonin norepinephrine reuptake inhibitors. And these antidepressant can elevate the symptoms of stress. However, they cannot reverse the suppression of the anti-tumor immunity. But there are other drugs which are available, such as mifepristone, that is an antagonist of the glucocorticoid receptors. And these drugs, when combined with the chemotherapy or tumor vaccination or PD-1 antibody-based immunotherapy, we do see that it can rescue the symptoms of stress by increasing the total locomotion, as well as reduce the level of stress hormones, cortisol. And also it can rescue the efficacy of chemotherapy and tumor vaccination, as well as the PD-1 based immunotherapy. Okay, we have a lot of upcoming questions. So role of microbiome. So the question is raised by Emanuela Cosini. And the question is about the role of microbiome. Do you know how germ-free animals respond to stress? Indeed, it's a very interesting question because germ-free animals respond to stress completely differently compared with these animals raised in the facility of SPF. There are already some reports about this point, and it's an interesting question, needs further investigation. So which are the bacterial or virus contributing to the stress phenomenon? Which are the, beneficial bacterial strains or viruses that can elevate the stress responses? Which is remaining a good question. Do you think mucosal immunity? So it's the question. Next question is raised by Face Oddity. Thank you for a very good presentation, of course. Thank you for your listening. Do you think mucosal immunity plays a vital key in reducing the impact of COVID-19? It's absolutely a very important question because we are right now in the process of analyzing the immune profile or the immune context in the GI tract as well as in the lung. So the systematic approaches will be applied to analyze different cell populations, including the innate cell population, such as ILC and MATE. So this is, of course, interesting question remain to be answered. And thank you for the suggestion. And the next question is by Adinasia. How to I think it's connect. It's how to connect stress that change the good or bad bacteria in the stomach in terms of COVID-19 infection. Well, in my opinion, that first we need a thorough investigation of what are the so-called good or bad bacteria. First, we need to know them. And second, I don't know whether there's a way to culture these bacteria by the culture mixed technologies. If it is possible, we can do that and perform adaptive transfer or further identify the important factors such as the beneficial metabolites or the detrimental ones to enhance them or to supplement them or to block the synthesis of these metabolites. This is another possibility. But sometimes I think that if the patient's already in very severe stages, it may be too late to use the microbiome adjustion strategies. Well, upcoming questions. So, then how is the virus stimulating immunity? Do you think stress affects cholesterol? Okay, the question is raised by Army Nasha. Do you think stress affects cholesterol rate and immunoglobulin concentrations in COVID-19 patients? I think it is absolutely an interesting question to investigate. And we do have some samples collected from patients, but the problem is that we need to always rule out the possibility that the patients receive medications such as dexamethasone, which can elevate the symptoms to some extent. And I think that the application, which also leads to a very similar questions, the application of the dexamethasone or other synthetic glucocorticoid is beneficial for patients. But I mean, we may also think about the possibility that the prolonged utilization of glucocorticoids or the large dose of the glucocorticoid, maybe it have some detrimental effect on the antiviral immune responses, which of course needs further investigation. A very lovely question proposed by Ursula. The question is, data shows that lactobacillus is reduced in the GI and the helicobacter is increased. Will fermented foods in the diet help COVID patients? Well, this is a question that obviously I want to know. If we can only take some food as how to say, the compensatory strategies to enhance our immune responses by adjusting the gut microbiome, it will be very interesting and beneficial, easy and practical. So another question raised by Sanyo Aurora. Please comment if the asymptomatic people do shed enough virus and which can transmit to the contacts as good as the symptomatic individuals. Well, this really depends on the viral load and of course the route of infection. And of course, I don't have enough experience in answering this question in a very professional way, but it is also an interesting topic that needs further discussion and investigation. Another question raised by Shamina says that, do you think any model on how physical activity and positive thinking action can moderate or improve the effects of stress? Well, I think that I have seen a lot of videos in interviewing the people who recovered from the COVID-19 diseases. And lots of them share the same comments that when they were in a very severe symptomatic period, they always give themselves encouragement and force themselves to fight for the loved ones, to keep breathing and, to keep hope. And I think that this positive thinking will really help with this antiviral responses or somehow elevate the inflammatory responses, which give yourself a chance to recover from this COVID-19. Thank you. Thanks a lot for all the audience who participate in this discussion. I think I'll take additional 2 questions and perhaps that's all of today. So the question proposed by Miyazaki, I think perhaps you're from Japan, and it had the questions showing, with the finding that perceived social support in self-experimental cases make the Th1, Th2 balance, Th1 dominant, is it likely to affect the COVID-19 infection as well? Well, I think really a balanced responses, including both T cells and B cells, are important. The Th1 and TC1 responses are important to, particularly cytotoxic T cell responses, are important to eliminate the infected cells. However, the Th2 will help the B cell responses. Also, I think really a balanced T cell responses and B cell responses will be beneficial. And others. A question raised by Lilina show that some people develop a mild disease and others a very severe disease, and genetic factors influencing the different immunological responses. Well, of course, the genetic factors may contribute to this point, such as the MHC class molecule expression, because the MHC may determine which are the dominant peptides, the viral peptides that can be recognized as antiviral antigens. Okay, perhaps that's all for today. Thank you very much for attendance and your interest in this very interesting topic. If you have further questions, you're very welcome to contact me through my email box, yuting.ma@163.com. And I think perhaps that's all for today. I need to contact the chair of the session. Hello? Yes. Thank you all very much for your time and interest. Stay safe and good luck to everyone. Bye-bye.